Okay, welcome to week 6. So, last week we talked about proteins, we talked about the structure of the proteins. So, today we are in this week 6, we are going to talk about proteomics. Basically, we are going to learn about all the tools which are available on the NCBI website, and we will explore the Scie, which is on the Swiss Institute of Bioinformatics. We will be learning about all the computer tools there, and you will learn various things in the field of proteomics. So first of all, I would like to show you there is a protein data bank, which is basically a principal repository for protein structure. So you can go to the protein bank and you can study the structures of a lot of proteins, and you can study the various and the functions of the various proteins there. So this is the protein data bank, which is a kind of great wealth if you want to study various proteins. And today the very first thing, so as we know, in the last week we studied the structure of proteins at 4 different levels. So today we are going to learn a few protein tools, proteomics, in which first of all we are going to talk about homology modeling. And then we are going to talk about how we can find different domains and motifs in the proteins, how we can find where in the cell the protein is localized, and how to find if the protein is present in the membrane of the cell, what region of the protein is in the membrane. So the very first tool we are going to learn is the homology modeling. So basically, homology modeling means to make a model of protein by using homology. So basically, like I give you an example. If you're working in a lab and you come across some protein which is nobody knows anything about that protein. So if a scientist has to study that protein, then in the lab, if you're doing the bench work, it can take few years to learn about that protein. So because the laboratory techniques are very laborious, and they need a lot of resources, they need a lot of time, so maybe years to study the protein and come up with its structure. And when you know its structure, you can come up with its function. But what is homology modeling? It's a kind of a bioinformatics tool. By using this tool, what you can do, there are some tools. We will be using the tool on Scie website today, which is the Swiss Institute of Biotechnology. So by making use of this tool, what you can do, you can in this tool you can enter the sequence of your protein. So the primary sequence you need to know. So if you know the primary sequence of the protein, which means you know the order of the amino acids, there will be a window where you can enter that, and then that tool is going to make the model of the protein. So it is going to make the model of the protein, which means that it is going to tell you the three-dimensional structure of the protein. And if you know the three-dimensional structure of the protein, then you know a lot about the protein, because the function of the protein is determined by its structure. It is determined by its three-dimensional structure. So when I talk about the three-dimensional structure, I mean, if you go back and look in your last lecture, we talked about the structure of the protein, which is at 4 different levels. The primary structure, which is the order of the amino acids. Then the secondary structure, when the chain folds, it can be alpha helix or beta pleats, beta sheets. Or if the chain folds more, then it attains a tertiary structure, which is because of the interactions between various R groups. And then if a protein is made of more than one polypeptide chain, then how do those fully folded peptide chains, they interact with each other, gives the quaternary structure. And quaternary structure will be only if a protein is made of more than one polypeptide chain. So this model, the homology model, is going to tell you the three-dimensional structure of the protein. And if you know the three-dimensional structure of the protein, you can predict its function. And you can also predict with what other kinds of proteins this protein is going to interact. So in the cells, basically these proteins, they interact with each other and they activate each other, and this is the way these proteins function. So if we know the three-dimensional structure of the protein, you know the function of the protein, you can predict it. I mean, it's not that it's a bad informatics tool does not give you the 100% exact answer, but you can make predictions, and then you can validate those predictions, which means you need to do some lab work, but your time will be, a lot of time and effort will be saved, because bioinformatic tools can give you, it can show you the directions, and you can definitely work in that directions and validate those predictions. So if you know the structure, you have a prediction for function, and also you can predict with what other proteins that protein is going to interact, and maybe if that protein is involved in some kind of in some kind of disease, you can also design some drugs. So it can go up to that level. So but how does this tool does that? It is going to tell us the structure of the protein. It is going to make the model of the protein on the basis of homology. So when you tell the system, when you tell the tool that this is the sequence of my protein, it is going to go and find the similar proteins, whatever the proteins are in the database, and then it is going to find the protein with which it is most homologous, and that protein with which this protein is most homologous is called the template protein. So in this kind of modeling, the most important thing is to find a template, because if template is wrong, then your model can be wrong, and you're not going to do anything. The tool is going to do everything, but you need to know how to use that tool. That's what I'm going to teach. So structure of a protein may reveal the information about the biological function of a protein. So if you know the three-dimensional structure of the protein, you can predict its function, you can predict with what other kinds of proteins it's interacting, and you can even design the drugs. So most of the time, three-dimensional structure is a good predictor of protein function. Therefore, protein structure prediction is a very important part of bioinformatics with important effect on medicine and pharmacology. So this is the homology modeling, or it's called the comparative modeling, which we are going to do. So you're going to learn how to do that, and by which you can predict the structure of the protein. So these are the computational methods for the protein structure prediction. So predicting the protein structure by using computational methods. So why do we need computational approaches? Because when we do all these things in the lab, as I said, it takes a lot of time, it is very laborious, it involves a lot of resources, and sometimes even if you use everything, sometimes there are so many problems in experiments, things don't work out. But on the other hand, bioinformatics tools, they save your time, they save your resources, and it gives you predictions. It definitely increases the efficiency of your work. And how does these models work? So basically, as I said, this tool is first of all going to find the template. So template recognition is the first step, and the template will be the most homologous protein to your protein, your unknown protein, and it is going to look in the database. It's going to compare with all the other proteins and going to find the template, and then it is going to align your protein, your unknown protein, with the template, and then it is going to determine what are the structure, what part of the proteins they're conserved and which parts are variable regions. It is also going to look for side chains, and then the refinement of the structure, and in the end, once all these things are done, the alignment is done, these conserved regions are found, the variable regions are found, side chains are found, and then the structure will be refined and it will be validated. So these are the steps in the modeling. In this, basically, as I just said, first of all, first step is template recognition, and then is the alignment. For alignment, basically, BLAST can be used, and then they are aligned, and then the regions which are same and which the conserved regions and the variable regions can be found. So after initial alignments and finding structural conserved regions among templates, we choose the final template. So there will be like the inside the tool, when it is going to look in the database for the proteins, there will be few proteins which are homologous to the your protein, and then it is going to make the comparisons between the various proteins which are all candidates, which means there can be like 5 or 6 templates which are homologous to the unknown protein, and then it is find for the final template with which it is most homologous. Basically in the homology search, it is basically the pairwise alignment which is used, which means the two proteins are mostly compared, but multiple alignments can also be used, which means your protein can be compared with multiple proteins too, but it's mostly pairwise alignments. So this is basically just the same thing which I am saying again and again. So after doing alignments and finding structurally conserved regions, which are abbreviation is SCRs, we align the unknown sequence with the aligned reference proteins with the knowledge of structurally conserved regions. So these conserved regions, they are made use of, and with the help of these sequences, they are aligned. After finding the suitable template and aligning the unknown sequence with the template, assignment of coordinates within the conserved regions is done. So this is the way how the this tool is making the model. So basically the model will be made by the tool. You just need to know how to use the tool and how to analyze the results. So these are some of the tools which can all make your models of the proteins, but we are going to use this one. We are going to use this Swiss model. Okay, so these are all various tools. Okay, so in the lecture I'm just going to give you some information about these tools, and in the lab, which is your computer lab number 3, we are going to actually use these tools. So second part is, first part was to make the 3-dimensional structure. The second part is to find protein domains and motifs. So first of all, what are domains in the proteins and what are motifs? Now domains is a region in the DNA, in the protein. It's a region in the protein which can work independently, and it can be responsible for some special function which is done by a protein. So it's a region in a protein which can actually function independently and can perform a function. That is a domain. Motifs, they are small or short conserved regions in proteins, and they're typically 10 to 20 amino acid residues. They can be they're very short. So domains are longer, domains are bigger, motifs are small. So motifs are like small short sequences in the protein which are usually conserved, and they can be sites for like phosphorylation or some other chemical groups can be attached. So domain is a region of DNA. It's longer than the motif. So So domain is a region of a protein that can adopt a 3-dimensional structure, and it can perform a function. Like the example of domains are zinc finger domains, immunoglobulin domains. So which means these are domains in the protein. So there can be 4 or 5 domains, and they are performing some special function. And motif is a short conserved region of a protein which is typically 10 to 20 amino acid long. This is example of some of the domains in the protein, like zinc finger, immunoglobulin, EGF-like. These are different regions in the proteins, and they perform some special function. Like here you can see in this slide, you can see these red parts. These are all domains. You can see in this protein it has such a big domain here. So this protein this is extending along the length of the protein. There can be domains which can be like little shorter also. There can be the domain can occur 1 or more times also. So here is example of a protein which has these 2 domains. The protein is called methyl CpG binding protein. It has these 2 domains. It has MBD domain and TRD domain. What is MBD domain? It is a methylated DNA binding domain. So which means this methylated DNA binding domain, this is the part of the protein which is responsible for binding of this protein to the DNA. And this is the transcriptional repression domain. So this protein is basically transcriptional repressor. So what are transcriptional repressor? Transcriptional repressors are the proteins which reduce the transcription. As is the process by which the information in DNA is converted into mRNA, and that mRNA information in mRNA is converted into protein, that is translation. So there can be, these are the two stages in protein expression. The first stage is transcription. So there can be some proteins which can act as transcriptional repressors, and there can be some proteins which can act as transcriptional activators. So this is a protein which is a repressor protein, and this is the domain which is transcription repression domain. So this part of the protein is responsible for the repression of the transcription, and this part, because if it is a repressor, if it is a transcription repressor, which means it needs to bind to DNA. So this part of this protein is responsible for binding of this protein to the DNA. So it is performing, so this domain is performing a function of repression of transcription. This domain of the protein is responsible for binding to DNA. And if you do the blast of this protein, and it has been found that there are various other proteins which also have this kind of domain. This MBD is the DNA binding domain. Okay, so this is, we can also use European Bioinformatics Institute, which is EBI, offers many key proteomic resources. Various proteomic resources are at this resource also, but we will be using Scie. Basically, we will be doing all the proteomics tools at the SIB, which is Swiss Institute of Bioinformatics, and it is also called Scie. So we will be doing everything in the computer lab. So this is what we are going to do. This is Swiss. So we are going to use Scie. This is a proteomics server. So it's expert protein analysis system. So you will be finding all the tools here, but you have to go under the proteomics. So we will be using this Scie. So to find the domains and motifs, we can use this tool, and you will find this tool on Scie. It is called ProSite. So ProSite is a computer tool by which is a proteomics tool by which you can find the domains and the motifs in the protein. So there you have to enter the sequence of your protein, and it is going to tell you what kind of domains and motifs are there in your protein. The next question is protein localization. If you want to know, suppose this is your cell. Inside the cell there's a nucleus. The proteins, they can, and this region is called cytoplasm, this is nucleus, and this is the plasma membrane, this is the membrane. So protein can be anywhere in the cell. Protein can be on the membrane, it can be embedded here, then some part of the protein is outside, some part is crossing the membrane, and some part is inside the cytoplasm. The protein can be here in the cytoplasm also. The protein can be in the nucleus also. So how to find the localization of protein? By using bioinformatics tools. There's a tool called PSORT. This is what we are going to use today. PSORT for finding the localization of the protein. And the next thing is to find transmembrane region in protein. Now what is the transmembrane region of a protein? So as I just said, like you can see here, if this is your cell, and if you have a protein which is on the membrane, which means when any protein is on the membrane, some part of that protein will be outside the cell, some part will be crossing the membrane, and some part of the protein will be inside the cytoplasm. So transmembrane region of the protein will be the part of the protein which will be crossing this membrane. So if this is the membrane, so here like it's a phospholipid bilayer. So basically transmembrane region means the part of the protein which is crossing the membrane. So they are the regions in the proteins which span the membrane. So how to find which region of the protein is um, crossing the membrane. For that we will be using this tool, which is called TMPRED. So this predicts the transmembrane regions. But what is the principle? How does this tool finds the region of the protein which is crossing the membrane? It is on the basis of the membrane. If this is the plasma membrane is rich in amino acids. It is rich in, it is made of phospholipid bilayer. Okay, so phospholipids, you can just imagine, phospholipids are a kind of lipids, and this membrane is made of phospholipids. Okay, and the phospholipids, they are bipolar in nature, which basically means they have they are phospholipids. They are amphipathic molecules. They have polar heads and non-polar tails. Okay, polar heads, they are hydrophilic, which means they love water, and hydrophobic tails, the non-polar tails, they are afraid of water. So the center, the core of the membrane, this part of the membrane is all made by the non-polar tails of the phospholipids. Okay, so just to explain you in a simple way, you can just imagine that this is the membrane and it is made of some kind of lipids, and those lipids, they have polar heads and they have non-polar tails, but the interior of the membrane is made of non-polar part of the phospholipid. And anything which is polar in nature, it loves water. Anything which is non-polar in nature, it is afraid of water. So it means that when I'm saying that the core of the membrane is made of non-polar tails, and non-polar tails are afraid of water, and they are called hydrophobic. So the interior part of the membrane is hydrophobic. The interior part of the membrane is afraid of water. You can just think like that. Now, any protein which has to be embedded in this membrane, in this membrane, that part of the protein, so it's an extremely important point here, the part of the protein which is going to be embedded in the membrane has to be made of hydrophobic amino acids, because you know that proteins are made of amino acids. So the part of the protein which is crossing the membrane has to be hydrophobic, because the interior of the membrane is made of non-polar tails and they are hydrophobic in nature. So how does this tool predicts the transmembrane region in protein? It is all on the basis of hydrophobic amino acids. So this tool, which is called TMPRED, it is going to predict which parts of the protein can cross the membrane on the basis of the presence of hydrophobic amino acids. So this is the basis of this tool. So these are the 4 tools which we are basically going to learn today. So this was the information I wanted to give you. That's all we need to know of the theory part, and then we will be talking about all these softwares in the lab.